instructor for Dr. Byron Quinn's Natural Science Biology 1 course at Langston University, and I've compiled a series of review videos, and this happens to be the last one, Chapter 15, where we discuss biotechnology. Okay, Chapter 15, we talked about biotechnology, cloning, goats in biotechnology, plasmids and their significance. We discussed Dolly the sheep, electrophoresis, and PCR. So let's jump in. So, biotechnology in general is using the technology to solve life's problems or to improve upon things. So, your genetically modified food, the cool stuff scientists can do usually falls in the category of biotechnology. So, in this case, they isolated the gene that the firefly uses to light itself up. You guys, most of you all have seen a firefly before, so it's able to light up, and that's caused by a particular gene. And so scientists were able to take that gene out and insert that into the tobacco plant, and it glue in the dark as well, just like the firefly, firefly, does when seen in nature. Another principle of biotechnology is cloning. So that has been in the news. I know you guys see it in the news. This particular horse is not necessarily this horse's child, but it's actually an exact clone of this horse. So it can be used in terms of livestock. If you have an award-winning horse or race horse or something like that, you can take an exact clone of that horse, and that's what happened in this particular figure. Another important, interesting thing in biotechnology is the use of goats, and that's significant because at Langston University, we are known for our goat research. And what the interesting thing about goats is that a particular firm called GTC Biotherapeutics, they were able to take genes of interest and put them inside of the goat. And what ended up happening was that the milk from the goat contained the particular proteins that the biological firm was interested in. And so what they ended up having were these goats producing milk that contained the proteins that could actually prevent blood from clotting. And so that was one interesting use of goats in biotechnology. Another important thing to remember is plasmids. Now, plasmids are little circular pieces of DNA that are found in some bacteria. And remember, bacteria fall under the group of prokaryotes. Bacteria and archaea, there are prokaryotes. Well, they're useful in science, especially in biotechnology, because they contain these things called plasmids. Now, plasmids, again, are these circular pieces of DNA. And what's interesting about these circular pieces of DNA or plasmids is that they replicate and divide outside of the normal bacterial genome. So they're separate from the bacterial genome. They're kind of just there. And so we're able to exploit these in the following slide. So for instance, let's discuss plasmids in biotechnology. We have a bacterial chromosome that contains the plasmid, and we have a particular gene of interest that codes for a protein. Well, what we can do is we can take that particular gene of interest and isolate that. And then we can snip it out using restriction enzymes and put that into our plasmid. And then once we have our gene of interest incorporated into our plasmid, we call that recombinant DNA. Then we can insert that back into the bacteria. Now remember from chapter, what chapter was that? I want to say chapter 9, where we discussed mitosis. Well, remember, bacteria in our prokaryotes don't undergo mitosis. They actually undergo a process of replication called binary fission. This is just review. We already discussed that in chapter 9. If you remember, binary fission occurs extremely fast. It's just when the one cell splits into two. And so this is useful to scientists because once we have our recombinant DNA, it is able to replicate extremely fast, and we're able to actually trick the bacteria into producing the protein of interest that we were interested in the first place. So this is a really cool way to use plasmids in biotechnology. So you can see how that works. And you've all heard of Dolly the sheep. If you see Dolly the sheep right here, is a white-faced sheep, and its surrogate mother was a black-faced sheep. Well, there's no way that a black-faced sheep can make a white-faced sheep. And so that was one reason why this black-faced sheep was chosen as the surrogate mother for Dolly. Because Dolly being a white-faced sheep, there was one way to prove that Dolly was actually a clone and not this black-faced sheep natural born offspring. So let's see, take a look at how Dolly was cloned. So what happened was they took udder cells from a white sheep. This is Dolly's real mother right here. And they were able to isolate the DNA and put that in the egg cell of a black-faced sheep. Now remember, the black-faced sheep is just the surrogate mother of Dolly. It is not Dolly's natural mother, it just carried Dolly. So they were able to take the DNA from the white sheep, Dolly's real mother, put it in the egg cell of the black sheep. Now the egg cell's DNA was inactivated, so it's just the DNA from the white sheep. And it was implanted into this black-faced sheep, or the surrogate mother, and the surrogate mother ended up giving birth to a white-faced sheep, Dolly, which is actually this particular sheep's exact clone. And so that is how Dolly was cloned. Well, two techniques that we discussed, that we briefly discussed in Chapter 15, Biotechnology, is electrophoresis and PCR. Now, PCR is a technique that scientists use to amplify genetic material. Now, one thing that you need to realize is that usually when scientists are able to get samples, their quantity is scarce. And so we have used a technique called polymerase chain reaction, or PCR, to amplify the amount of genetic material that we have, so we have more to work with, more to do our samples and experiments on. And another technique that we use commonly, scientists use commonly, is electrophoresis. Electrophoresis exploits the fact that DNA has a naturally negative charge on it, so it's going to migrate towards the positive end, because opposite charges attract. And so what you're able to do is you're able to sort your DNA fragments based on the sizes. And so the longer fragments, of course, are not going to travel as fast as the short fragments, just like, excuse my colloquial terms and bad example, but for most parts, a fat person isn't going to run as fast as a skinny person. And so the skinny person, or the short DNA fragment, is going to migrate further down the gel than a longer fragment. And if you have known samples, you can compare that and say, oh, okay, well, these happen to be the same sample, or so on and so forth. So you can make conclusions from that based on the fragment size. So those are two techniques that we discussed in Chapter 15. PCR used to amplify genetic material, and electrophoresis used to sort DNA based on their size, the fragment size. Alright, well that covers Chapter 15, guys. So good luck on your final. And remember, biology is 98% hard work and dedication and 2% genius. It was great working with you all this summer. Make sure you review the videos 1 through 15. Took some time making these for you, so you can't say that, oh, I don't need to go over that part. You can pick and choose the ones you need to go over. And I hope you guys do well on your final tomorrow. That's all, folks.